The overall goal of this procedure is to propagate and detect an infectious clone of Maedi-Visna virus that expresses green fluorescent protein. This green fluorescent protein has been cloned into the gene coding for DUTP-ACE in the Maedi-Visna virus genome. The infectious clone is contained in two plasmids. First, the two plasmids carrying the Maedi-Visna sequences are cut with XBA1 restriction enzyme and ligated. Second, sheep choroid plexus cells are transfected with the construct. Finally, Maedi-Visna virus infected cells are detected by fluorescent microscopy and flow cytometry.
Hi, I'm Stefán Jónsson at the Valgerdur Institute for Experimental Pathology at the University of Iceland. Today we will show you a procedure for propagating and detecting an infectious molecular clone of Maedi-Visna virus that expresses green fluorescent protein.
The molecular clone is contained in two plasmids, P8XSP5, EGFP, and P67OR, of 12 kilobases and 4.5 kilobases respectively. Cut equimolar quantities of the two plasmids, namely 4.4 micrograms of P8XSP5, EGFP, and 1.6 micrograms of P67OR with XBA1 and ligate before transfection. When ligation is completed, incubate the ligation mix at 60